Okay, so we're starting this last session of talks at this meeting, and it's at the mitochondrial pharmacology session. And our first speaker today is Eleonor Aasander Frostner, and she's going to be talking about towards a treatment for mitochondrial disease, current compounds in clinical development. Whenever you're ready, thanks.
Thank you. So good afternoon, and thank you very much for this opportunity to say a few words about primary mitochondrial diseases and current clinical drug development within the field. I work as a communications officer at Abliva, a small biotech company in Lund, Sweden, and also a little bit as lab manager at a research lab at Lund University. Just a small, to disclose that I am an employee of Abliva and I own shares, and Abliva has a commercial agreement with Ouroboros. So, primary mitochondrial diseases are a group of severe and inherited diseases that are caused by either mutations in the nuclear DNA that affect the mitochondria or in the mitochondrial DNA itself. They affect about 125 persons per million in the adult population. And they are often multisystemic disorders, and many tissues and organs are affected at the same time. And often in organs that require a lot of energy, like the brain or the skeletal muscle or the heart. The most common symptoms and the most debilitating ones are fatigue and myopathy, which is muscle weakness. And the fatigue is not the normal tiredness that you and I can feel after staying up late, like today. Patients have told us that even a simple thing as taking a shower can be very exhausting for them, just by the temperature regulation and the muscles they're using. It can affect the rest of their day. In children, the most common symptoms are not neurological. So the effects of the disease can arise in any organ at any age and with varying severity. And historically, they have been very difficult to diagnose. You mostly rely on checking the family history and also using specific diagnostic criteria that take into account the clinical findings, biochemical findings, and imaging findings. And they often involve the need to take a muscle biopsy, which can be very invasive. But the knowledge about these patients is increasing, and also things are moving forward with the use of whole genome or exome sequencing, and also biomarkers is an emerging field. But there's no cure, nor any pharmacological treatments available, and current standard of care is basically taking care of symptoms as they arise. It's like putting out fires without ever reaching the source. Dietary supplements are widely used, but without any evidence of efficacy in controlled clinical trials. And you can also manage the diseases with avoiding different external stressors. And current therapeutic development is within many different fields, but I will focus here on drug development of small molecules that are in or will soon be in clinical development from the industry side. So these molecules act through many different mechanisms of action, and we have chosen to divide it like this for this talk. So we have metabolic reprogramming and mitochondrial biogenesis. We have energy replacement and substrate enhancement, and also regulation of ROS. We start with the reprogramming and biogenesis. We have the compounds that are PPAR delta agonists, PPAR delta is a nuclear receptor that promotes fatty acid oxidation and also can regulate genes involved in biogenesis. At the bottom we have our compound, Abliva's compound, KL1333, which is a NAD plus NADH modulator. So one of the effects of mitochondrial disease is a depletion of NAD plus and accumulation of NADH. So KL1333 normalizes this ratio, and by increasing the amount of NAD plus, you can also stimulate pathways that are involved in biogenesis. To the right, we have a guanylate cyclase stimulator, and guanylate cyclase is part of the nitric oxide signaling pathway. When it comes to energy replacement, we have our second program, NV354, which is the second generation of cell-permeable succinate prodrugs. And one of the first generation prodrugs is available for research use from Ouroboros. NV354 is in development for bypassing complex 1 deficiencies, which is can be found in Leigh syndrome, for instance, which is a pediatric disease. To the right, we have a drug candidate that targets also a pediatric disease, TK2 deficiency, which is a mitochondrial DNA depletion syndrome. And this drug candidate contains nucleosides for DNA synthesis. When it comes to regulation of ROS, we have 2 vitamin E-based redox modulators that act through either thyrodoxin or glutathione. And to the right, we have a cardiolipin targeting compound. And cardiolipin is a phospholipid specific to the mitochondrial inner membrane. And this compound binds to cardiolipin and protects it against ROS. So what do these clinical trials, what is evaluated in these? So many look at fatigue and myopathy, either through a specific walk test or symptoms questionnaires that the patients answer, or functional tests for muscle endurance. And some look at the neurological symptoms, for instance, through looking at the frequency of epileptic seizures or brain perfusion. And some of them target the pediatric diseases and look at they're assessing motor milestones. So I'm happy to say that a lot of these compounds are in late stage development in phase 2 or phase 3. Phase 3 is usually the last step before a potential market approval. And there are also some new compounds coming into early development. So hopefully we can within a few years have a new or a first treatment for the multisystemic disorders. So with that, I'd like to thank my co-authors, and the full manuscript is available as a MitoFit preprint. And I'd also like to acknowledge the many patient organizations that are very important to the patients, of course, but also in drug development. And it's a good collaboration. So that's it. Thank you.
Thank you very much. We've got time for some questions. Anybody?
The question is, what was the criterion to use PPAR delta agonist instead of PPAR alpha agonist? We know, you probably know, that PPAR alpha agonists like bezafibrate or fish oil are not toxic, are well tested, and people consume this as, like candies.
Yeah, I think that PPAR delta was the only one that that had been shown to be involved in mitochondrial biogenesis specifically. And
No,
No?
PPAR alpha regulates beta oxidation, gene transcription, all of them. And besides, also all the transporters and enzyme synthesis for cholesterol. PPAR alpha, I think. Well, that's the question.
yeah, we can probably check with that company. So yeah, can't answer that.
Eleonor here, very nice work. Yeah.
Where are you?
Yeah,
There?
we have eye contact. Very nice work. I was wondering about your KL133 compound, where it modulates NAD over NADH levels.
Yeah.
There's a lot of work on this NAD supplementation. Different compounds have been tested, but they're never that successful somehow. Can you elaborate a little bit more what your compound exactly does and why that's different from the ones that have been tested already?
Yeah, well, KL1333 is a substrate for NQO1. So it NQO1 uses KL1333 as a substrate, and it uses NADH as an electron donor, and then KL1333 gets reoxidized by cytochrome C in a redox cycle and can work very efficiently if you compare to let's say CoQ10 or
So
Adiponone.
it doesn't increase your total pool of NAD and NADH, but it rather changes the ratio.
Yeah, exactly.
Okay.
Mm.
Okay. Can I ask a follow-up question? Or yeah. Just a quick follow-up question. The work in this field always attracts a lot of attention from doping agencies, because that could also increase exercise performance.
Sorry, I have a little trouble hearing you.
So, try it again.
Yeah.
The work in the mitochondrial diseases field gets a lot of attention from the doping field too, because it could increase exercise performance, your drugs.
Yeah.
Do you see any of that? Because I saw there was some muscle endurance test in your measurements, but
Yeah.
can you comment on that, whether that is a
Well,
possible?
yeah, personally, I actually don't know, but I mean, it's a possibility. I mean, I think a lot of people want to take advantage of anything that makes the performance better. So yeah, that's probably true. Yeah.
But you don't have any data on whether it does or does not improve performance in a healthy state as well.
Yeah, well, our compound has. We had a phase 1A/B study with a small cohort of patients, and it was just 10 days of dosing, but we could see, encouraging trends towards improvement in fatigue and also myopathy, and also the improvement correlated with exposure of the drug in the blood. So, but it's just a small
Very
cohort.
Very
Yeah.
encouraging. Thanks. Good
Very
stuff.
I'm actually interested in your NV354 succinate prodrug.
Yep.
So this idea of bypassing complex 1 impairments is obviously out there for a while. I know there's a patent on a trisuccinyl glycerol, but it's very, very difficult to make. So can you tell me a little about what this is? Maybe I missed it. I was a little late. But what is the structure? Is it succinate itself, or what do you mean by prodrug?
Well, I don't think that we have revealed the full structure yet, but it is succinate with small side chains, and it's I think it's a type of N-acetylcysteine bound to it. So.
And orally active. If you feed it to mice, then you get higher levels of succinate in blood.
Yeah, exactly, and it reaches the brain as well. So it's encouraging for Lee patients. Yeah.
Thank you.
Okay, I think we're running really nicely to time. Thank you very much for a nice talk.
Our second speaker is Maija Dambrova from Latvia. She will be talking about mitochondrial metabolites, acylcarnitines, therapeutic potential, and drug targets.
Yeah, thank you very much. So, acylcarnitines, a little bit of chemistry. Those are molecules, esters, that are formed from moiety of carnitine and fatty acids. And fatty acids can be of different shape and kind, and usually they are classified as medium-chain acylcarnitines, long-chain acylcarnitines. And at the moment, in the 5th edition of Human Metabolome Database, there are more than 1,000 molecules reported, and acylcarnitines are a class of compounds measured in untargeted metabolomics studies. We got interested in acylcarnitines as metabolites emerging from fatty acid beta oxidation pathways in mitochondria, but acylcarnitines are also produced in peroxisomes, and peroxisomal oxidation, beta oxidation is very important for very long-chain fatty acids, also long-chain. After peroxisomal part, mitochondria can take care of so there are no more very long-chain fatty acids, hopefully, and mitochondria can go on with further metabolism, which ends with ATP production in the mitochondria. A very important molecule in this pathway is malonyl coenzyme A, because it regulates activity of CPT1, carnitine palmitoyl transferase 1 enzyme, which synthesizes acylcarnitines for long-chain acylcarnitines for further transport inside mitochondria. And this is important pathway for long-chain acylcarnitine formation and long-chain fatty acid metabolism. This all goes on very nicely if conditions are physiological, if a person is metabolically flexible, and if there is enough oxygen for normal mitochondrial bioenergetics. Since those are important metabolites for metabolomics, it means that they appear in the bloodstream. We have done preclinical studies which show that for the long-chain acylcarnitine pool in the bloodstream, heart is the main organ producing long-chain acylcarnitines. Skeletal muscle is more important for medium-chain acylcarnitine appearance, and liver for short-chain acylcarnitines. We did this in correlation, by correlating concentrations in tissues and also different kind of bioanalytical measurements. It should be noted that concentrations of long-chain acylcarnitine, and here is an example, and in following figures I will use palmitoyl carnitine. Its concentration in tissues and also blood plasma changes during the fat-fasted state switch in both in humans and also in preclinical models. What happens in the fat state? In fat state, when we have more glucose in the circulation, insulin signaling starts, activates malonyl coenzyme A production, which makes CPT1 less active. Long-chain acylcarnitine concentration decreases, and we get less fatty acid flux and also less fatty acid oxidation. And especially for clinical samples, it is very important to understand at which stage samples are collected. Are those from fasted persons or fed, and especially when insulin effects and insulin sensitivity is tested. Long-chain acylcarnitines accumulate a lot in case of inherited fatty acid oxidation disorders, and any of enzymes shown here result in high levels of long-chain acylcarnitines, and those patients exhibit cardiac complications, also arrhythmia, sudden cardiac death, which is difficult to treat, and at the moment it's kind of diet and not very much pharmacological treatment related to long-chain acylcarnitines. By accumulation, and this is like for the notion that long-chain acylcarnitines are not just a transport form, but they are signaling molecules themselves. They can affect ion channel activities. They influence phosphorylation level of insulin receptor, ACT phosphorylation level, PDC complex activity in the presence of long-chain acylcarnitines. We get more ROS production in mitochondria. We have tested palmitoyl carnitine and seen that at physiologically relevant concentrations, it decreases OXPHOS respiration rate, cell viability, and it can stop, in fact, isolated heart at low micromolecular concentrations, just like that. We developed a pharmacological agent called methyl-GBB, with which one can decrease acylcarnitine concentrations both in tissues and in blood plasma. Our experiments in isolated heart and also in vivo shows that in the case of decreased long-chain acylcarnitines, we have lower level of infarct size. And also we developed a genetical model which is characterized by lower carnitine level and also decreased long-chain acylcarnitines. It's knockout of trimethyllysin hydroxylase, very live animals. And again, just without any pharmacology, we get smaller infarct size. There are pharmacological means by which one can modify concentrations of long-chain acylcarnitines, and those include also doping drug meldonium, but many others. In the Bioblast preprint, we have a table with some of very used drugs that have those effects, and also many clinical trials include measurements of acylcarnitines and L-carnitine, and those are related to both to fatty acid oxidation diseases, the diabetes, but also mood disorders and many others. We have drafted a review about acylcarnitines as biomarkers, also naming possible therapeutic potential of changing the levels, naming drug targets, and analyzing clinical trials. And I would like to thank colleagues, also Christiana from Ouroboros and my team in Riga, colleagues in Sweden, Germany, and also David Wishart and the Human Metabolome database team that added a lot of new information into the database, and we hope that our work will be important both for mitochondrial pharmacology, but also for metabolomic studies by providing information about physiological and pathological effects of acylcarnitines in preclinical research and also in clinics. Thank you.
Thank you, Maija. The floor is open for questions.
Very interesting talk. You haven't mentioned anything about neonatal screening. I mean, in the Western world, in all industrialized countries, we have full quantitative acylcarnitine spectra of virtually every newborn. Have you thought about potential applications in tackling all these dehydrogenase deficiencies in beta oxidation that are diagnosed there? Because the patterns are really not just non-targeted metabolomics, as you mentioned, it's really quantitative data with single-digit CVs.
You are absolutely right, and this is the topic where acylcarnitines are the markers for already in clinics, and this is kind of accepted. What is less accepted is insulin sensitivity, also physical activity, and so on. And I think that the problem is this measurement in fed and fasted state, and also we collaborate with clinical colleagues, and for them routine is to collect samples in fasted state. There are publications, sometimes inconclusive, about long-chain acylcarnitines, but when we ask authors, it turns out that they don't know in which state the samples have been collected. And then the problem is that, in fact, if there is some person, especially in the emergency, with high level of glucose, you don't know if this person is fed or this person is diabetic. And the treatment, and also, like maybe if we remember talks from morning today, so glucose is when it saves the life, maybe when it doesn't have any effect. But yeah, you are absolutely right. And because of that, and those tests for inborn errors, since this is kind of already clinical method, I think that measurements of different types of acylcarnitines could be affordable also for other biomarker types and other pathologies. And short-chain acylcarnitines are used as food supplements. And again, is it like biochemically correct or not? Many more measurements are needed. And this is both metabolomic assays, but those are also simply pharmacokinetic assays of some like acetylcarnitine, propionylcarnitine, and there are studies showing effect, studies not showing effect. One more thing, okay, I can let you ask questions, but no, I also heard several presentations during this meeting talking about coenzyme, long-chain coenzymes and fatty acids. The thing is that for coenzymes and fatty acids, there are binding proteins. For acyl, and especially for long-chain acylcarnitines, it's a bit of question mark. At the moment, we are running a structural biology study to find what happens with long-chain acylcarnitines, and it looks like they are dangerous and appear in the bloodstream when binding proteins are very occupied by coenzymes and also fatty acids. But still, I think that analytical chemistry during the last 5-10 years have been really improved, and they are ready for clinical samples. And also we in Riga are ready to analyze some well-characterized samples and maybe advertise using of palmitoylcarnitine, octanoylcarnitine, and different kinds of those long-chain acylcarnitines, and also not long-chain acylcarnitines in mitochondrial measurements, in addition to glucose-derived substrate used in the protocols that everybody knows here. And we promise to provide data from Mitopedia and new protocols for nice pages of Bioblast and so on. Thank you.
Just a quick question. You talked about fasted and fed. Are you aware of the diurnal rhythm on acetylcarnitine concentrations? And the second is the fasted state the standard to assess acetylcarnitines?
No. In fact, to study insulin resistance, acylcarnitine should be taken at the same time points and as during the glucose tolerance test. and they change, like glycose goes up, long-chain acylcarnitines go down, and this kind of shows insulin sensitivity. And in fact, you can start to measure changes in the decrease of acylcarnitines, maybe even during the pre-diabetes sooner than this is with glycose. So, but again, this is something that we should test more, and animal experiments are animal experiments and human are human, and yeah, and then sports part also.
Thank you very much. Thanks.
Thank you.
So next we have a series of 10-minute talks. The first of these is from Omar Torres-Quesada from Austria, and he's going to be talking about kinase perturbations redirect mitochondrial function. Thank you.
So thank you very much for the kind introduction. So first, I would like to thanks to the organizer to give me the opportunity here to present my work. And second, I would like also to apologize in the name of my colleague Erich Stefan, because he would present software after me. He thought, but he couldn't manage to come today. So I will fuse the talk so that I can bring you a broader overview of the kinase inhibitors and the work that we are doing. So the talks are entitled Kinase Perturbation Redirect Mitochondrial Function. And shortly, so first I would leave you a short introduction about mitochondrial kinase networks and kinase pharmacology. In the second part, I will show you a little bit of the profiling of kinase inhibitors that we are doing in the context of the MitoKin project, just underline the effect on cell culture media and showing an example of profiling of the kinase inhibitor PFK 158. And then in the second talk, so I will also give you an idea of the system that we are using in the lab also to predict kinase drug sensitivities and also activities. So mitochondria are not isolated organelles, so they are center places where the plethora of signal transmission signals are converging. So the signals are coming from the membrane and they are transmitted signaling molecules to this organelle and also from other cell compartments that they call in protein kinases. So protein kinases phosphorylate proteins and regulate its function. Also phosphorylates mitochondrial proteins, and this phosphorylation usually exerts a strong regulation of the activity of these targets. And of course, also in the mitochondria, controls functions like, for example, mitochondrial bioenergetics, like ATP production, also mitochondrial bioenergetics, biogenesis and dynamics like fission and fusion, and cell fate like proliferation, differentiation, and also cell death. However, these mitochondria, so these kinase networks, they might become aberrant. That means that due to mutations and also kinase aberrant signaling, they produce a deregulated function also in the mitochondria, which usually it's connected with several diseases, like, for example, Parkinson's disease, diabetes, or cancer. In cancer, it's mainly one of the major strategies. It's use small molecules targeting kinase activities. And here you can see already, so they were designed a lot of kinase inhibitors which target these kinase activities. And because but usually they are not, so the mechanism that which inhibitors affect mitochondrial function are not well understood. So kinase inhibitors represent the major cancer drugs used in pharmacology. So they are around 158 kinase inhibitors inhibiting and from 660 kinase of the human kinome. So the property of these kinase inhibitors is they have a broad spectra that they can inhibit several kinases, even hundreds, but also they are inhibitors, they can inhibit a single kinase, representing are really attractive molecules for cancer therapy and drug development. However, because the mechanism of action, so they are not too specific, it can also trigger off-target responses that at the end that relied in so side effects or non-desirable side effects. So in our project, it's the mitokin, which is coming from mitochondrial and kinases. We, and this is a project together, a collaboration from the university and Ouroboros. We aim to identify kinase pathways that are via mitochondrial function relevant in cell proliferation and in cancer development using unsystematic profiling. So in our systematic profiling, so we use, so we combine several strategies. So first, we use as a readout kinase activities and drug efficacies using, for example, kinase conformation reporters that I will talk about later. And second, we also combine with the impact of the metabolite compositions. So third, we also analyze the effect of mitochondrial function using high-resolution respirometry. And finally, we also analyze the effect of these kinase inhibitors in cell proliferation and survival. So because we are mainly cell biologists, we work with bidimensional culture. Our first thing is if we can somehow recreate the more physiological conditions for our experiments. And so we compare actually the effect of cell culture media in our experiments, and we took the classical media that are, for example, RPMI or DMN, and we compare with the newly formulated human plasma media, so the effect of cell proliferation and mitochondrial function. As you can see here, so the composition of the classical media, it's quite far away of the human plasma, seeing that they are lacking many of the polar metabolites that they are present in this human plasma, but also they have a completely different concentration. So that's the reason that usually they do not mimic the composition, and we wanted to address if these differences are relevant in cell proliferation and mitochondrial function. So first, we analyze the effect of classical, the newly formulated human plasma media in cancer cell proliferation as compared with the classical media using two standard 3 cancer cell lines. So we use a colon cancer, a breast cancer, and a melanoma cancer cells. And as you can see here, we observe that the human plasma media affects the the proliferation profiles of these cells in a cell-dependent manner, indicating that HP element affects cancer cell proliferation in a cell line-specific context. Second, we analyze the effect on cell respiration of cells growing either in classical media like RPMI or DMN and this newly formulated media. And as you can see from the living cells, so we observe that there is also the media influences really the mitochondrial coupling state, showing that, for example, from no effect in the mitochondrial coupling state, like you see here, to decrease of the mitochondrial intrinsic uncoupling to the enhance of the mitochondrial density, indicating that human plasma media modifies mitochondrial respiration also in a cell line in the specific way. So third, we also wanted to address if the cells, when they are grow in either classical media or human plasma, so the drug sensitivity was affected. So in this case, we grow the cells in both medias, and then we expose them to the tyrosine kinase inhibitor sunitinib, which is actually a drug which is using in the clinic against cancer and inhibiting 160 kinases. So surprisingly, we could find that when the cells were exposed were grown in human plasma and then exposed to the sunitinib, we could identify just mitochondrial partial discoupling that was not observed when the cells were grown in classical media, indicating that HPLM also influences mitochondrial drug sensitivity. So then, after identifying the effect of cell culture media also in drug sensitivities, we decide to apply our mitochondrial profiling and kinase profiling in using another specific kinase inhibitor. This is the PFK 158. That actually it's an inhibitor which inhibits one of the glycolytic enzyme, the PFKP3, and it did not overcome clinical trials actually because of these high side effects. So we decided to analyze the effect of kinase activities and also cell respiration upon treatment of these inhibitors, as we could observe an effect in kinase conformation using the King-Kong reporter that I will speak later, and mitochondrial function in living cells really in a dose-dependent manner upon 1 hour treatment of this inhibitor. So here, the experiments, the high-resolution experiments show that there is a strong mitochondrial discoupling both in flux upon the treatment with this inhibitor. Also then, afterwards, we wanted to see if the mitochondrial pathways are affected with this inhibitor. And surprisingly, we could identify that PFK 158 produces a strong dysfunction of the end-linked respiration, probably related with pyruvate metabolism on pyruvate transport, but also effect damaging the mitochondrial outer membrane, indicating that the oxygen fluxes and the fluxation ratios would at the end produce a strong mitochondrial failure. These effects, they couldn't be explained just for the single inhibition of a glycolytic enzyme that would suggest that the PFK 180 has really a strong off-target effect, inhibiting or producing a mitochondrial failure, probably for other different non-expected ways. So then, just to take home message for this part, so I hope that I convinced you that interlinkage of kinase mitochondrial networks is relevant in human diseases. Then physiological cell culture media modifies cancer cell proliferation, mitochondrial respiration, and drug sensitivity compared to classical formulation, and mitochondrial profiling of kinase inhibitors can predict off-target effects. It can be important, for example, in the preclinical evaluation of cancer drugs. So then, this I will give you at the end. So then, for the second part, we also wanted, because of the interesting effect of the kinase inhibitors, to also generate somehow a tool to track patient mutations and also lead molecules driven by the alteration of kinase activity conformation. So as I mentioned before, the development of novel kinase drugs, it's really something important in pharmacology because many blockbuster drugs are used for cancer treatment. And actually, out of these 158 kinase inhibitors, 71 are already FDA-approved drugs. However, because there are a really huge amount of clinical trials, so more than 1,500 ongoing, so they really represent a huge marker site. However, so many of the drug candidates fail in clinical trials, so that means that new technologies for the validation and research of the drug candidates are needed. And this is what we designed, so that the King-Kong Biolabs designed. So we designed this King-Kong biosensor platform. So it is a cell-based reporter assays to analyze kinase conformation. This reporter is based in the renilla luciferase, so an enzyme that usually naturally produced by luminescence. And the system consists in basically fuse to the full-length kinase protein fragment 1 and fragment 2 to the S, N, and C terminal part of the kinases, and then expresses in cells. So here you can see that in this reporter, it can be in the open conformation. That means that the fragments are not in close proximity and they don't produce bioluminescence. However, when the kinase switch to a more inactive, so more close state, the fragment gets close into proximity, and then this leads to producing of a bioluminescence signal. This is interesting because these changes in between closed and open conformation, they are in kinases really dependent of, for example, drugs, post-translational modification, but also mutations that can be predicted or can predict drug efficacies. And this is actually the benefits of this method. So, for example, usually for the traditional approaches for drug reproportional drug screening, so they are based in vitro models that often are restricted to not the full-length proteins or more kinase domains. They are complicated multi-step protocols. So with the King-Kong biosensor, so they are cell-based. That means that they could also predict toxicities. They analyze full kinases. They are just simple, so it's just luciferase and emitted light, real time, and also they are patient mutation specific. So this is, there can be, so here you can see how this is the case of kinases that they were assessed, or the drugs that they were in the kinase family. And in the King Kong report, actually, we could extend this kinase report not just for the typical tyrosine kinase, but also for other families that they represent the dark kinome. Then here, to finish the last part, so I would like to show you just an example of how come with the kinase, so the King Kong platform can be used. And this is probably, you will see, this is the most prominent or the most important kinase pathway in cancer. This is the MAP kinase pathway. This is the major pathway which is deregulated in cancer. And this is because there are many mutations that, for example, in the receptors and also several oncogenic proteins that they lead to cancer. And I would like just to focus in one of the biggest or most important points, which is BRAF. And BRAF actually mutations in this gene leads to several types of cancer, but in the case of melanoma, there is a prominent mutation, which is the V6 and the E mutation, which leads with uncontrolled cell proliferation, and at the end, so the melanoma, poor prognosis. So we use our King Kong platform to track the activity of kinase drugs that they were designed against the specific mutation. And here you can see that in the wild type, we couldn't see any effect because these drugs are mutation specific. But in the V6 and the E mutant, we could see how most of them, several drugs, they trigger the reporting to the more close conformation, so they inactivate the kinase, but not all of them that are actually, some of them they are in clinical trials, they didn't show this activity. So also, we try to extend this observation not in melanomas, but also in lung cancer, because in melanoma, so this mutation is around 92%, but in lung cancer, so there are many BRAF mutations that are also related with cancer, but one of the strategy is somehow use these BRAF inhibitors as a therapy, but the problem is usually that the effect in these mutations are not well understood. So we introduced these all mutations in our reported, and we actually could see different drug behaviors depending of the mutation background. And this we could confirm in cells, so in lung cancer cell models, and we could correlate somehow the activities of in the King Kong reporter and also the proliferation, indicating that King Kong platform could be also useful to predict drug behavior and efficacies. And this is just coming to my end. So this so we also extend this mitochondria platform, the mitochondria. I already explained you, so the PFK, and we tried so to also understand the effect of several kinase inhibitors in the PFK53 pathway, but also we are now trying to identify activators for Parkinson's disease and related kinases involved also in the mitochondrial quality control and mitochondrial dysfunction. So then at the end, so I would like to thank especially Edward Stefan and all the group of the Institute of Biochemistry to also collaborate with us. And especially I would like to thank to the Mitokin Lab, with we perform mainly all the measurements. And also I would like to especially thank Erich and Ouroboros for a strong support, and the Tiroler Krebsforschung Institute for also supporting the place and the way to perform our research. Our collaborators, our funding agencies, and of course, thank you very much for your attention, and I'm happy to take questions.
Thank you very much, and thanks very much for stepping in for Stefan as well. Do we have any questions?
Thank you. So I have a question on your King Kong sensor. Maybe can you show the slide, or is that not allowed?
Yeah.
The bioluminescence one.
This one?
Yeah. So you have this sensor. Is this reversible, or
Yeah.
is this a one-way reaction?
No, it's reversible. So it's always depending of the conditions. So the reporter can be from active, so open state to the closed. But then, for example, if you add a drug which drivers the change to the other conformation, then so the reporter can also be so go back to the other conference because the interaction between the two fragments is just structural, it's not covalent. So that means it can be just depending of the proximity that is related with the conformation, can be either dissociated or associated without reversible.
It cannot associate by coincidence. The F1 and F2.
I mean, this is a good question because one question that might be if this interaction might be also between not just between the same molecule, also between other molecules. So we assume that the interaction between so extramolecular interaction, they are less pronounced than the interaction between molecules. So, but of course, it's also possible that there are, for example, in case of dimers or something, that there might be also interaction between fragments in different molecules.
Okay, and I guess the length of the linkers is important as well.
Yeah, exactly. Yeah, okay, because the linkers, of course, they are big tags, so they could also affect the function of the protein.
Maybe time for a very quick question with a very quick answer, if anyone has one. Oof, you have to be quick with your hand as well. Just down here.
Yeah, very interesting. I think at the end of your first talk, I guess, at the highest dose of your kinase inhibitor, it looked like after you added rotenone, it went above like the regular ETS. So do you think that's affecting complex 2 inhibition prior to adding rotenone?
So you mean from the sunitinib?
Yeah.
We don't know, so because we try to also go to mitochondrial pathways, but we use one protocol with a specific substrate combination, and we couldn't see, so we saw this coupling produced by the inhibitors, but we couldn't see clear effect in one of the complexes. However, we saw when we analyzed the fatty acid oxidation that the fatty acid oxidation was reduced upon the treatment, so with the inhibitor. So that might also explain the effect. So.
Thank you very much. We can continue afterwards as well. Thank you.
Thank you very much.
Thank you. We go now to two last talks, 5-minute talks. The first one is by Andrea Magri from Italy. He will be talking about modulation of mitochondrial respiration in ALS cells by hexokinase-based peptides, a novel therapeutic approach to fight neurodegeneration. Thank you.
So thank you so much. I would like to thank the organizer of this meeting for giving me the opportunity to be here today and present our data. So, neurodegenerative disorders are a group of pathologies characterized by the accumulation of misfolded protein within the neurons and the aggregation at the mitochondrial level, as observed, for example, for a beta peptide in Alzheimer's diseases, alpha-synuclein in Parkinson's disease, and a specific mutant form of the antioxidant enzyme superoxide dismutase 1 mutants in amyotrophic lateral sclerosis. Most of this interaction occurs through the presence of VDAC1 on the outer mitochondrial membrane, which is also known as mitochondrial porin and represents one of the most abundant protein of the outer mitochondrial membrane. Due to its beta-barrel structure, it allows the passage of ions and the passive diffusion of small metabolites, which include metabolites up to 5 kilodalton, such as pyruvate, succinate, but also nicotinamide cofactors, and especially ATP, ADP. So it plays a key role in the regulation of mitochondrial bioenergetics. It has been shown in ALS mice that the deposition of SOD1 G93A mutant on VDAC1 dramatically affect metabolic exchange through the mitochondria and limiting the ATP availability and initiating the mitochondrial dysfunction, which is another feature typical of ALS and the other neurodegenerative disorders. So based on this observation, we queried whether the use of small peptide targeted to VDAC was able to reduce aggregation at the mitochondrial level and to restore the VDAC1 functionality. So with this purpose, we decided to use as a template the amino-terminal domain of hexokinases 1. Hexokinases is the first enzyme of glycolysis, of course, but it is also one of the most important and abundant interactor of VDAC1 in physiological condition. In fact, in neurons, for example, about 80% of hexokinases is bound to mitochondria through a direct link with VDAC. So the resulting peptide, called NHK1, was able to interact with VDAC in a two-system component and to modulate VDAC1 conductance as the entire hexokinases can do. And if added to recombinant VDAC1 or purified mitochondria, it prevents aggregation of SOD1 G93A. But however, we recently tested our peptide in a cellular model of ALS, NSC34 cells, which stably expressed SOD1 G93A mutant. We observed that the high hydrophobicity of the peptide was enough to observe an uptake of this peptide, at least partially, within the cells. And this correlated with a significant reduction of cell death in this model, which was linked to the expression of SOD1 G93A mutant. Also, nice result we observed in using respirometry. In fact, it was previously demonstrated that the expression of SOD1 G93A in the same model by the group of Elisa Calabria correlated with a significant decreasement of oxygen flux and an increasement of daily respiration. With the peptide, we observed a partial increasement of specific flux, a decreasement of daily respiration, and an increasement specifically in the coupling efficiency and in the net flux related to the OXPHOS. By Western blot on the mitochondrial fraction, we observed that the peptide correlated with a decrease of about 20% of the SOD1 at the mitochondrial fraction, and surprisingly, we observed also an increasement of VDAC1 expression without disturbing the endogenous level of hexokinases. So overall, the increasement of VDAC and the decreasement of SOD resulted in a decreasement of about 70% of the toxic aggregation VDAC1 SOD1. So in conclusion, these data, even if they are just preliminary, just demonstrated that the VDAC1 could be a valid pharmacological target, and at the same time that this strategy to use interfering peptide targeting to VDAC1 could be also a valid strategy to fight mitochondrial dysfunction during ALS. I would like just to thank my group in Catania University and all of you for the attention. Thank you.
Just in the nick of time, 1 second left. So, questions? Oh, sorry, no questions.
Questions afterwards, if you like. So our last speaker this session is Zuzana Sumbalova from Slovakia, and she's going to be telling us about ubiquinol supplementation accelerates the recovery of mitochondrial health of patients with post-COVID-19 syndrome on mountain spa rehabilitation. Thank you.
Thank you very much. So I am happy to share with you the results of our research on COVID-19. So in January 2021, we measured the mitochondrial respiration in platelets of patients after acute COVID-19, and we found a deficit in complex 1-linked capacity of oxidative phosphorylation and electron transfer. And electron transfer. The concentration of CoQ10 was reduced in platelets, and this study was published in Bratislava Medical Journal. So in May 2021, we moved our lab to Mountain Spa Resort, to sanatorium of Dr. Gur, located in high Tatras at altitude about 1,000 meters. And this is the sanatorium specialized on curing respiratory diseases since 1898. And this is also the first sanatorium in Slovakia providing a special program for patients with post-COVID-19 syndrome. We use short protocol for respiration of platelets, focused just on complex one-linked respiration. The patients with post-COVID-19 syndrome 3 to 6 months after COVID-19 arrived to sanatorium for 16 to 18 days of mountain spa rehabilitation, and there they randomly divided into two groups. MR groups was just on rehabilitation, and MRQ groups was on rehabilitation plus supplementation of coenzyme Q10 2 times a day, 100 milligram in the form of ubiquinol. Blood samples were taken first morning after arrival and last morning before departure. And at the same time, 6-minute walking test, exercise dyspnea, oxygen saturation, and clinical symptoms were recorded. In 8 patients of supplemented group, additional samples, blood samples were taken after additional 12 to 14 days after coenzyme Q supplementation at home. Lungs function and clinical symptoms improved in both groups after mountain rehabilitation, as seen by increased distance in 6-minute walking test and decreased exercise dyspnea. And more clinical symptoms disappeared in the group supplemented with CoQ10. In mitochondrial respiration, we could see that complex one-linked capacity of OXPHOS and electron transfer was reduced significantly reduced in both groups, and these parameters slightly improved in group on mountain spa rehabilitation, and slightly improved and significantly improved in the supplemented group, and even more improved after additional days of CoQ supplementation. Concentration of coenzyme Q in platelets was not initially different from control and increased markedly after supplementation. And there was a positive correlation between all parameters of respiration with CoQ10 concentration in platelets, and the increase in OXPHOS and ET capacity correlated with the increase on CoQ10 concentration in platelets. And there was a trend to positive correlation between the increase of intracellular CoQ10 concentration and the increase in lung function test. So our data show significant role of supplemented coenzyme Q10 in regeneration of mitochondrial health in patients with post-COVID-19 syndrome. So I would like to thank all patients and collaborators and Comenius University in Bratislava and Kaneka Pharma Europe for a donation of ubiquinol. Thank you very much for your attention.
Thank you, Zuzana. Thank you, everyone. This concludes our session. Please, Verena.
So just a general announcement for this social program this evening. So we will have the guided tour for the Black Nights. So we will meet at 6 directly in front of the Hofkirche. So you have the address in the program. Maybe you type it in the Google Maps. It's quite close to here. So this is a permanent exhibition. And we have a guided tour with 3 groups, a German group and 2 English groups. That's why you should have all, in the meantime, already some dots on your name badges.
These are necessary to assign you to the groups that they don't get too big. If you don't have the dot yet and you stay for the discussion now, I hope so, then afterwards you can go to the registration desk. Faye will hand out the correct color dot to you, and otherwise in front of the Hofkirche you should come to us that you get your dot. Okay, thank you. And afterwards is the celebration then.
Hi. Well, thanks everyone for joining our session to discuss the future of BEC. I'm Luisa. I decided to present some aspects, some frequent questions that are commonly asked, so we can all start on the same page, and then I'm looking forward to hear the opinion of all of you. Well, one aspect that many people ask is about indexing. People ask when BEC is going to appear in PubMed, for instance. Just to let you know, there are two ways to be indexed and appear in PubMed. One is through PubMed Central, PMC, the other through Medline. And they have some minimum requirements. So, for instance, they require to have a registered ISSN. We obtained this for BEC at the beginning of this year, after we had last year one issue with 5 publications. But they also have other requirements. One of them, for instance, is the minimum of 25 peer-reviewed articles for PMC. For Medline, it's a bit more strict, I think 40 peer-reviewed articles. So we hope that now, with many articles that we received now in conjunction with Bioblast 2022, that we are going to get its numbers soon and then be able to be indexed and appear in PubMed. We also got questions about the directory of open access journals. It's also a way of indexing. And they also have some minimum requirements, again, the ISSN. And they also have some requirements that journals must have a minimum of 5 articles per year. And for new journals, they require to have a minimum of 1 year of history or at least 10 articles for newly launched journals. And there are also other aspects that might keep us from being able to be included in DOAJ so soon. So, for instance, they required that the proportion of publications in which editors or reviewers are one of the authors must not exceed 20%. And so far, most of our publications include either some of our associate editors or also reviewers. And about the Creative Commons licenses, this is the license that we apply for BEC. So in this aspect, we are already matching the requirements. Another common question is regarding impact factors. So the traditional impact factors are the ones that are kept by Clarivate Analytics. And this ratio is based on counting of citations. Also, many other impact factors are based on citation counting. And they also have some requirements so that you can get the impact factor. Of course, you have to have at least more than 2 years history to get this 2-year citation that is used for their calculation. But you also have to be indexed in the Web of Science, and they have some criteria for inclusion of journals, some quality criteria. Once a journal enters, it enters an emergency sources group, and then just after some time it can be included in the group that will have the impact factor calculated. And there are also other impact factors nowadays. So there is Timago, age index. They're all based on different calculations, but all based on citation counts. And there are also some other aspects, like the directory of open access journals, for instance. They say they do not approve the use of impact factors to evaluate the quality of a journal, but on the other hand, they ask for the journals to keep only the Clarivate Analytics, the classical impact factor, on their web page if they want to be included in DOAJ. But the impact factors are nowadays more and more discussed if this is really a good measure of a journal quality. And we would like to bring to the discussion the DORA. So this is an initiative to change the way to evaluate science in general, not only journals, but also careers for career progression and so on. And one of their recommendations is to decrease the use of the impact factors. They have several problems. They were originally created as a way to help librarians to select journals to purchase for their libraries. So they are not exactly made for evaluating quality of science. And they have several recommendations. So BEC signed DORA, I think earlier this year, or it was already last year, now I don't remember. Yeah, and perhaps Eric would like to add some in this aspect of impact factors and citation counts. So if you would like. Thank you. I'll sit here.
Well, thank you, Luisa, for this overview, and thank you for staying with us even for this final discussion on the future of BEC. I would not like to give too much additional comments here. I can reflect the history of BEC. Why did we start such a crazy idea to launch a journal when we know that there are too many journals in the world? There is a logarithmic increase of the number of journals. When I was teaching at the university environmental protection, I showed the increase in scientific journals correlating with the extinction of species. We are even environmentally concerned scientists are not really in practice environmentally concerned. We are talking about the world population increase as a threat, but we are not concerned that the world increase of publications, which we cannot cope even in digesting, is not a threat. The more publications I have, the better I am. And the higher the impact factor. Now, if you observed the citations in the presentations, usually one cites Nature, Science, Cell, PNAS, and other journals do not appear in proportion to the impact they have on the science, I think. So we started in the spirit of DORA as contributors to abstracts or even mitochondrial physiology preprints, the mitophyt preprints, or now even through the process of BEC peer review, you realize that we changed the format of citations. So right now we still do citations in publications like in the last century, as if the internet didn't exist. We do not need page numbers, we do not need journal names, we need authors, we need the title, and then the link to the internet. And all this fuss about formatting, is it page numbers total, is it a dot, is it all this formatting is so unnecessary, and the length of the citation so unnecessary. It is really doing the job of today with yesterday's tools. So the formatting, when all you need is really a link. That may be not in the slides, in a presentation. You cannot really digest or read such an internet link. There you can memorize more easily a journal. So I think for the purpose of presentations, that's it. But for publishing, there is no need to use the tools of yesterday. So that is what, just in the spirit of DORA. Why is this in the spirit of DORA? It is not emphasizing the Nature, Science, Elsevier, Springer journals. they have the highest profit margin of any business probably in the world. Maybe I'm lacking information, but the profit margin of, say, a real journal like New York Times or so is 17%. Elsevier, Springer, 42% profit margin. Why? We are the journalists, we pay the journal. New York Times has to pay the journalists. Okay, we do even the formatting. We do the job. The quality control is again done maybe with a 10% discount for our next publication. But again, it's done by us. And then the journal is bought by the university library. And the state, or we as a population, as taxpayers, pay again. And 42% is the profit margin of these paywall journals. Now, the term of paywall journals is ambiguous. I call paywall journals any journal that asks the author to pay, that asks the reader to pay, that asks university libraries to pay, so where essentially public money is paid to give access to governmentally funded, usually governmentally funded research. So I think the business of science journalism is one of the most corrupt businesses in the world. Elsewhere bought for multi-million euros from Robert Maxwell. He was the first icon to run scientific journals as a business. It was Pergamon Press. Multi-million euros went into the purchase by Elsevier. So you can imagine how much profit they make to afford buying such a thing. So the business strategy is very easy for these paywall journals. They ask, do you want to become an editor? Oh, yes, for a journal. Do you want to be on the editorial board? Oh, yes. And maybe with a bottle of champagne at the evening, or a cruise besides a conference. I mean, Robert Maxwell rented a whole hotel besides a conference and invited the future journalists, the future editors of the journals. So he started so many new journals, and the university libraries, with some good articles in there, had to purchase them. So it started really as a business not to help science, but to exploit governmental resources. And we are the slaves of paywall journals. That is my perspective. And when we say there is no conflict of interest, well, we, in any of the Springer or Elsevier or other paywall journals, one conflict of interest there, it should be assigned as a paid advertisement. Now, JBC had always, I don't know, they still have, it's a paid advertisement. That is our no conflict of interest approach. Everything that we publish in these paywall journals is paid advertisements for our careers, for a PhD, for our next fund, for our pay rise, paid advertisements. So we are liars. And the liars are, number one, the paywall journals. So these reflections, I had time to think rather than travel during the lockdown. I was going deep into this literature by studying what is the advantage of preprints and then papers, what is really the statistics about the success of peer review. And I must say all my experience with peer review have been mixed. So a good peer review process improves your paper so much. It's amazing. You have worked maybe version 24, already 25, and then you submit and you think, now it's perfect. And a good peer reviewer opens your eyes for such a tremendous improvement. And in this way, we thank all of you who have helped as peer reviewers in the processes that we have now started with BEC. It's hard to get peer reviewers interested in peer reviewing a non-indexed journal, but therefore it is even more important to thank you for this collaboration. Thank the editorial board team to support us, and only with this science community collaboration can we ever make it into a good journal. Now I said I will not talk very long. You realize I'm a liar. This was just to give you the spirit why I said, hey, let's get together and do a Don Quixote approach, running against windmills. Let's have the fun of, we will see the Black Knights tonight, the knights that have run against many, many windmills, some of them successful, some of them not. Now, what is the future of BEC? Is it worthwhile to engage in such a journey? I'm convinced if we have illusions in our minds, then we stay a bit more aggressive and young. So it is an illusion to change the world, but let's do it nevertheless. So that is my little incentive, and maybe we had, I think, also those who have contributed already to the discussion on the website. So Marcus, maybe you want to give some comments and get us a little into the discussion. Sorry to take most of the time, but Marcus, please.
Well, first of all, thank you, Eric, for this honor. I mean, Eric just invited me to give a brief discussion about it. So my perspective, as I put in the back website, I think it's kind of different because coming from Latin America, the reality is completely different. I fully agree, Eric, that the market is corrupted for a long time from a paywall perspective, but I would add that for the open access business is even worse. And that's the point that I'll give you some numbers. In the last 10 years, the price of APC for the open access journals, for high impact factor journals, rose about 80% above the inflation in the US. No, open access. It's APC, it's not paywall. We can discuss which kind of paywall we're talking about. Yes, yeah, but this is open access through the Plan S. That's the problem. For the lowest impact factor, it rose just 26%. So my point is that the way that the open access is being pushed now is directly linked to our obsession with the impact factor. Indeed, this was calculated. There are studies that shown that the impact factor, the journal impact factor, correlates to about 30% of the APCs. So we think that if we don't change our culture, and I know that this is very hard because we are addicted to the impact factor, the problem will persist. I mean, it will change the business from paywall to open access, and the problem, I think, is going to stay. So my point is, maybe a start discussion on, we could do it, is which model of publication should BEC use? That's why I asked if it was golden open access or diamond open access, because I don't think that in the long term, and maybe in a medium or long term, this will be sustainable in my perspective, because as long as Ouroboros could support it, that's fine, but I don't think that will be sustainable in the long term. So maybe this will be a first point to discuss which model of publication we should adopt for this new journal. I'm not against the golden open access, but I'm against the commercial exploitation as being pushed by the Plan S, as it is, especially for the Latin American and many other countries in the world that the researchers cannot afford for the price of an APC of a journal, like a Cell or Nature that charges $10,000 for a paper. This is almost a year of salary of many researchers in many places of the world. And that's the main point. That maybe is the first point to discuss. I have many other points, but I don't want to give this further. But that is the main point for me.
Does this work? Yeah.
Thank you. Well, thank you very much, Marcus. I totally agree with you. If we use the usual paywall definitions, that's what you did. But I tried in my editorial for BEC to define paywall on many different other levels. So the open access and you pay behind the scene, I mean, that is also a paywall for the authors. So paywall is not just for the readers, paywall is for the authors, and I think that's what you have addressed. So with the number of publications per year, when you in PubMed just go for mitochondria and chloroplasts, then the daily publication means 100,000 euros we pay per day, just for mitochondria and chloroplasts. 100,000 euros unnecessarily lost to the scientific community to do productive work. And that is, I think, that is the paywall, not only for the readers, but for the authors. And then Elsevier does not disclose the contracts with the universities. And you referred to the 80% increase of the open access. Now, how much a small university has to pay for an Elsevier journal and how much one of the Stanford big universities, they don't disclose the contract, so we don't even know how much money goes there. So how is it sustainable? Yes. We thought we have in the past, before the lockdown, supported an open science approach with organizing conferences, and we asked for very modest registration fees. I hope you agree that this was now not overwhelming for you. And that was our support. Now, we did not run any scientific conferences for these years, and we thought we were extremely successful thanks to you. And we thought we give a little bit back, not in terms of losing money by advertising. We do constructive PR. I mean, we are very clear. We wanted BEC to be the journal of the Mitochondrial Physiology Society, but then legally that is a very difficult thing. We started it with project money. So in our next-gen O2K project, European Union funded, there was 50% of the 2.4 million euros was 50% for dissemination and for just manuals, etc. So dissemination, starting this journal, starting the website, was also funded within this next-gen O2K project to the company Ouroboros Instruments. So we cannot easily just say now it's the Mitochondrial Physiology journal. It is disclosure. It is very clearly run by Ouroboros Instruments, but in a completely open science way. So if you publish, if you submit, it is not O2K linked, of course, not by chance. There are many contributions using our technology. That is fine, but it's absolutely open and not restricted. So it has to be, I think, for legal and public funding or European funding reasons it has to be an Ouroboros Instruments journal right now. I mean, run by Ouroboros Instruments, but in an open science way. So how do we sustain this in the future? If we stay successful as a company, we can give like a little tax back to the scientific community rather than wasting our money with useless advertising. So it's a very open. I think if we are successful as a journal, if the scientific community accepts this, then it is linked to Ouroboros in a very positive way. So there is some scope of sustaining it. But if we are very successful, we will not be able to run this every month, something like this, okay? We do not really know what is the limits of this perspective of the BEC Bioenergetics Communications. We may change format. I think Johnny suggested as a possibility to run a biannual BEC conference. Yeah, I think we can do this next year already with the idea of the mitochondrial physiology conference linked to BEC action. I think that's a very good idea, and we may change the format. We thought we have an inaugural issue. Now that was too short notice and not possible, but maybe in 1 year we could say we have a number of peer-reviewed presentations, and we use the conference before going into publishing in BEC. It's already as a preprint, and then we use the conference to have more global open peer review, and then after the conference with the discussions, and the discussions could be even made available also as a part of the publication. So that could be a format. And this we can afford. But we don't even want to push to just high numbers of publications. This is a suicidal approach. I mean, can you read 8 new papers per day? That was 2 years ago. Now it's more per day than if we have MITO scope. So the high numbers, that's the living communication idea. Well, this you find on the website. So there are some ideas to not just contribute to the number of publications, but to the quality and to open peer review. Of course, we are not the only one, and many, many aspects that have improved in the scientific journalism now with open peer review, with many initiatives are good in the journals, in some journals. And we just want to put them all together, take the best out of what exists and add something new. So with this, I would like us to think about paywall from many perspectives. And the S initiative does not address this paywall, only the readership upfront paywall, not the backup paywall by the authors. So this is also non-acceptable, particularly for less privileged countries that yes, are excluded from a exactly 6,000–7,000 euro manuscript fee.
Yeah. Well, I'm not sure. One.
Well, thank you for letting me say a few words about BEC. I'm a very strong believer of this journal because I come from a university where we cannot actually get journals. Like I go on PubMed and I find something that I would like to see and read, but of course it's behind paywall. Now with the open access thing, it's better, but still we need this model to help us disseminate our work and also to read other people's work. So I was very excited to be part of this, and I really want to continue the collaboration, and I would like to see other scientists in the field. And not necessarily from this very nice group, but from other places as well, to contribute to help establish this journal. And establishing a new thing is always nice. It's always, you feel a sense of purpose, you feel a sense of achievement when you are part of this effort. So I'm very proud to be a part of this effort, and I would, even though it's still not indexed, and even though our universities, all universities will not count those papers when they look at promotions and all that, because they're not indexed. Even though that's what's happening right now, I'm optimistic that in the future this will be indexed somehow, and those papers will not go to waste. And so far I have 2 papers in the inaugural issue, and I'm actually planning to submit another one pretty soon. So despite all the obstacles that we're having, and I'm totally in for submitting another one and another one and another one. I really want to see this idea grow and grow. And thank you.
Any comments? My comment is, well, of course, we don't have the illusion that if you are supporting a PhD student, the committee requires impact factor, and there is no way to publish in BEC for a PhD student in most universities. So this is not our decision, but of course, in a way, we are running the system. What is the university? It's us. And we cannot blame the system without blaming us. It is part of us. But we are not able to do anything alone. No way.
I just want to add a comment. And it doesn't, I mean, this is a political, economical debate, and it's bigger and it's on the long term. And I agree with the BEC initiative 100%. I just want to say on the readership side, I hope everyone is aware of Sci-Hub. To pirate papers, and I think we need to use it more and to support Alexandra, I forget her name, but the Kazakh person who is under tremendous law in a pursuit, in a lawsuit in the US. And even at UCL, I use Sci-Hub because, I don't know, just out of principle. And I think maybe we need to pressure our institutions to stop paying the fees and use, anyway, pirating more, but it's an ideological question. I know not everyone will agree, but yeah, that's my comment.
Yeah, you mentioned we should try to influence the university libraries. Now, Johnny, you asked how can we, or how can you? Now, I'm not any longer part of the medical university faculty. I'm retired at the university, but how can you get a new journal without paying into university systems? I don't know whether any of you has here experience that, I mean, it is open access, so what should the university do? But you asked how can, maybe you reiterate your question.
My question was not that. My question was, how do we convince the university, the administrators, the vice president for academic affairs, whatever, that the papers that we are publishing in this journal that is still not indexed, but run by really well-known scientists, how do we convince them that this is true science, this is important? Because we have so many, we cannot count the predatory journals that are out there. And these journals so many scientists fall into their traps. And this is not something that anywhere closely resembles that. And that's my question. How do we convince them that this is really legitimate? That's why.
I think we are running out of time. I don't get a prize now for sticking to the program. We have to do some slight exceptions. Now, if you are not needing to go to your hotel, we have 20 minutes until we are almost outside of the door to assemble for the Black Nights. So I think this is the time to just thank you all once again for joining. Thanks to all the participants who have been active, either with posters, talking, discussing, joining the walk and talk, and having fun. I thank again our team very much. Even after the Black Nights is not over, we have then a celebration dinner. I hope all of you can join, or most of you can join. And if at the conference dinner there is not, the voices are, we don't have a microphone there, but certainly there I will thank you again and hope to see you soon at one of our events. The Mitochondrial Physiology Society event will be next year, end of June. Hope to see you there. And then in 2 years it will be a challenge, European Bioenergetics Conference. I'm not sure whether I can convince the committee that this conference, this is usually Elsevier, always Biochemical, Biophysical Acta, the invited talks. Elsevier is one of the paywall journals in the sense that we pay, somehow we pay. Now, if we would have a conference fee of 5,000 euros, which is maybe not Elsevier, but typically high-level journals ask easily with this 80% increase, 5,000 euros. If we would have 30 publications, 5,000 euros, well, we can, with 60,000 euros, we can at least have a good conference dinner. So I certainly work hard with the committee, but the committee is male-dominant, old people, not changing easily. But maybe if any of you has contact to any of the EBEC committee members, I'm on the committee, but I'm quite alone there. If anybody has friends from the EBEC committee, work on them that they consider maybe BEC for EBEC. That would be one goal. Why should we pay Elsevier with our conference fees? Now, maybe some of you know, EBEC conferences start always, EBEC is not a legal organization, it's just a bunch of crazy scientists, the committee that run EBEC. So if you run EBEC, we are running EBEC in 2 years, you have no budget, it's all on you. You can ask high registration fees, you can make a lot of money out of such a conference, which has been the case, that for 2 years you can run your lab after running EBEC. It was in the past. So if we pay with our conference fees for Elsevier, what a waste. So we have to work on committees, we have to work on university people, we have to change the system. Change is not easy, but let's do it together. Thank you very much for joining Bioblast 22. Verena, you have some last but not least announcement, and at the dinner we continue, of course.
No announcement. So you took away all the words I prepared for my little speech at the end. No. So I would like to thank you all for being here in the name of the team. I have to say we received lots of praise, and I want to say that we had the worst and the best case scenario at this conference. Worst case was that the main organizer was unfortunately not with us because she got sick. She was involved in every little detail of the conference, but she couldn't get the spirit from here. And that's actually the best case, so I couldn't imagine that it will be such a great meeting again. So after this long break, so last meeting we organized it with this size was in 2015 at MIP conference. It showed us that the team still can do it, and I hope that you bring the spirit to the celebration dinner tonight, to your colleagues at home, so that we can even activate more people for the next MIP school MIP conference in 2023, and for EBEC 2024, which will take place at exactly this venue. So thank you once more in the spirit of the team.